So you learned a whole lot about DNA, and you probably already knew that different people's DNA was different, right? We are unique because our DNA is unique. Um, and as long as we're learning all this stuff, I thought, why not take a moment to teach you how to read forensic evidence or to figure out the paternity of a child, right? Then you can get a job on the Jerry Springer show, I guess. Um, in your DNA SymBio lab, you're going to be learning about a process called PCR. With PCR, you could take a little bit of someone's DNA and then you can amplify it so you've got lots and lots and lots of DNA. That's by PCR. Once you've got lots and lots and lots of DNA, you can mix it together with an enzyme that will cut the DNA at a specific sequence. For example, if you've got a sequence AGT, um, I can't remember the exact ones. But if you've got a sequence that's like AGTA, let's just say it's not, it would cut exactly there, right? If it was AGGA, it wouldn't cut. So even if you and I make exactly the same protein for something, our DNA can be different. And if our DNA is different in a spot, then these particular enzymes will cut in a different area. So what would happen if you took my DNA from a crime scene and you amplified it in the lab, and then you cut it up in the lab? You would end up with some pieces of DNA, that are really small, and some pieces of DNA that are very long, and some pieces of DNA that are kind of in the middle. If you take all of that DNA that was mine and you amplified it and you cut it up, then you put it into this gel electrophoresis thing. Gel electrophoresis sounds really fancy, but what this is a material that's kind of like a thick jello, and you're going to put the different samples, like this might be mine, that might be yours, right? And then you've got big pieces and medium pieces and little pieces. Your pieces will be different groups than mine because your DNA is different than mine. So it'll cut at different sequences than mine. Okay? And then we're going to turn on an electric current, and the electric current is going to pull all of those pieces through the gel. Okay? And what's going to happen? What's going to happen is the small pieces are going to go, they're going to travel really, really fast. The middle-sized pieces, it'll be harder for them to do the little twists and turns, but they'll get through there pretty fast. And then the big pieces, sometimes the really big pieces don't even get into the gel. So then you'll have something that looks like this. This thing over here on the right, that's a real DNA fingerprinting gel. You won't have anything this complicated to look at. The ones that I give you will be kind of like hand-drawn, so you won't have to guess as to what lines up and what doesn't line up. So in yours, you won't have any of what are called controls, but you will have like victim, suspect 1, suspect 2, and then you'll maybe have a couple of samples from the crime scene. All right, let's try to read it. Now, remember, every column is one sample of DNA. So this is one sample of DNA from the crime scene, and you'll see it has a medium-sized piece and another medium-sized piece, but is larger. Here's another DNA sample from the crime scene, and it only has one piece showing, and it's quite small. Okay? Now, what we're going to do is we're going to compare it to the different people that are a part of this particular crime. So there's the victim of the crime and a couple of suspects. Great. Okay. Now this is probably the victim. DNA sample 1 is probably the victim of the crime. Do I know for sure? Not yet. This gives me like 85% certainty. If I want additional certainty, I would take the amplified DNA and I'd cut it with a different enzyme. The different enzyme cuts at a different spot in our DNA, but if this and this are the same DNA, then they will line up again. like if I cut it with a different enzyme, the pattern is going to look totally different, right? But my victim and my DNA sample number one will be the same every time. With different enzymes, the gel will look different every time, but those two lanes would always match, right? So that's DNA sample one. DNA sample number two is here. And it is compatible with DNA, with suspect number one from the crime scene. Now, do I know that DNA sample number two is from suspect number one? Not yet. Okay. When you get one match, you get a certain percentage certainty. And then when you cut it again, it gets more and more certain. I think by the time you cut it with 3 different enzymes, your certainty is almost 100%. But we do know from this one test, we do know that this is not suspect number two's DNA. So if the only reason we are holding suspect number two is because of the DNA sample, we will have to let him go because I don't know with certainty if it's suspect one, but I'm still going to hold him while I run some more tests. I know it is not suspect number two. All right. Now, let's do paternity testing. Paternity testing is the same up until the point that you're reading the samples. What do I mean by it's the same? What I mean is we got the DNA samples, we amplified the DNA sample with PCR, and then we cut them with enzymes, blah, 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 right? It's just how we read it because the child is not the same as the mother or the father, right? These are 3 unique individuals, and they will have 3 different DNA fingerprints. However, whatever little chunk of DNA shows up in the fingerprint, they either got from mom or dad, unless they have a mutation. Okay. But they either got from mom or dad. So we are going to always assume that the mother is the biological mother. By the way, family A, I always look at these pictures and I think someday one of my students is going to go, Dad, because these are just pictures off the internet. I didn't even put together these slides, so I don't know where they came from. But these are not real fingerprints, and this says nothing about this family. So please don't think badly of these people because of my stupid slide. All right. Okay. So we're going to look at the mother and the child. Now, the first thing we're going to do is we're going to check and see which bands of DNA did not come from the mother that are found in the child, okay? So bands of DNA, all right, this one did not come from the mother. That one right there did not come from the mother. Let's do it again. Okay, that one didn't come from the mother. And this one and this one, these did not come from the mother, okay? So they should have come from the father. But if you look, okay, these are the ones that did not come from the mother, great, okay. Let's look at which ones, did they come from the father? No, okay. So this man is not the biological father of this child, at which point Jerry Springer's show goes, ooh, okay. Let's do one more. Again, not this family, okay? That's just a picture on the internet. Don't put your pictures on the internet unless you want to end up in Tydell's lecture. Okay, so let's start with the son, right? I'm going to start with the son. Let's look at what bands are in the son that did not come from the mother. So this did not come from the mother. This one did not come from the mother. This one did not come from the mother. This one did not come from the mother. Okay, did they come from this man? Yes, 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 and yes. All right. It looks to me like this man is the biological father of the boy. Okay, now let's look at the daughter. That's ominous, right? You know the daughter is not going to match. Okay, don't just assume. Let's double check it. Okay, so this did not come from the mother. That one didn't come from the mother, but that's a little iffy. This one did not come from the mother. One of those didn't come from the mother. Okay. Does this man have those? No and no. All right. So this shows that this man is the biological father of the son, but not the biological father of the daughter. Okay. That's it. I will see you guys next week.